Good day. This is the Professor, CBMD PhD, and today's Thursday, July 20th, 2023. It is 3:35 and 53 seconds, a.m. Eastern Standard Time, and today's research, immune cells. So let's jump to it here. The scientific investigators and microbiologists say immune cells in single file, date July 17, 2023. Source, University of Wilkesburg. There is news from the immune system. Dendritic cells migrate to the network along the outside of blood vessels. Local cytokines keep the dynamic network stable. How do these networks form, and how are they maintained? For the long-lived macrophages, phagocytes, the answer is already known. They settle in so-called niches. These are environments of connective tissue cells that supply the macrophages with nutrients and keep them alive. These immune cells are essential for the control of immune responses because they are at the first line of defense of the immune system. They recognize foreign structures, take them in, and process them into a kind of mugshot. They then present the photo to other immune cells and trigger a specific immune response, for example, against pathogens or cancer cells. The special thing about dendritic cells, they only live for about a week, and during the time, they continuously migrate through the body's tissues. In this respect, it was clear that the classic niche concept would not work here. But researchers found a completely new concept for this, according to which three-dimensional cell networks can organize themselves. Dendritic cells orient themselves to the blood vessel and migrate one after the other along their outer wall, similar to children walking in single file. The blood vessel thus determined the three-dimensional arrangement of the cells. They wanted to understand how this process is regulated and how the cells manage to close gaps in their networks. Closing such gaps is important because otherwise the immune defense no longer functions optimally. The scientific investigators and microbiologists say it is due to a locally acting cytokine, the FLT3 ligand, that the dendritic cells always stay close together during their developmental migration. The cytokines are continuously and evenly produced, locally and consumed by the dendritic cells. If there are gaps in the group, more cytokines are available for the isolated dendritic cells. As this surplus speeds them up in their development and movement, it helps them to reconnect with the group. When the cells have moved up, they have a little less cytokines available, again due to competition from their neighbors. Accordingly, they slow down their developmental speed. These findings are, for example, important for cancer therapy. Dendritic cells have a high prognostic value for tumor diseases. The higher their abundance in the tumor, the better the prognosis for the patient. This is especially true after immunotherapy. Increasing their basic insights on dendritic cell biology will help them to restore the networks of these cells and tumors and thereby tailor optimal therapies in the future. Researchers' data so far is based on the analysis of lymph nodes from animal models. The team next wants to test whether the same principles of network organization of dendritic cells apply to all tissues and also in humans. And this is a Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.